חושב שהרבה אנשים נבהלו מהכותרת.
נבהלו, וואו. זה עצה טובה, לא? בהמשך.
לא, זה תלוי גם בקונטקסט, כי אני לא יודע, לא עשיתי פה סקר מאיזה תחומים אנשים מגיעים, אבל יכול להיות שיש פה אנשים שיודעים בדיוק מה זה value site.
הסקטור האמיץ.
אין לי מושג מה זה, אבל זה נשמע מגניב.
הופה.
הפוך על הפוך.
זה חלק for targeting bacterial cells.
אני נרשמתי דווקא בגלל הכותרת.
גם אני חייב להגיד.
And if you want some cookie research, this is the place.
אוקיי, אז אני חושב שאני רואה שאין עוד הצטרפות עדיין, מקסימום מישהו יצטרף באמצע, נכניס אותו.
Okay, great.
You can start, thank you.
Great, so we're rolling. Okay, so welcome everyone, welcome courageous audience. Please mute yourself. So my name is Ofer Prinz from the Faculty of Biotechnology and Food Engineering at the Technion. And in our faculty, and specifically in our lab, and under the supervision, of course, under Professor Ofer Prinz. In our lab, we really believe in combining all kinds of fields of knowledge, sometimes unpredictably, and sometimes a little bit crazy. So as a representative of this approach, I would like to welcome you to the exciting interface between someone who is not on mute, please mute yourself. Would like to welcome you to the exciting interface between microbiology, geology, and some nanotechnology. And to start to illustrate my motivation, I would like to illustrate a simple question. Imagine yourself ordering something nice over the internet. For sure, you would not expect your supplier to send thousands of packages around the globe in a desperate hope that one of them will reach you. Doubtlessly, he will send the right parcel directly to your address. Why then, when we suffer from some kind of illness, let's say gut infection, we flush our whole body with antibiotics, killing the good bacteria over the bad one, leading to the so-called dysbiosis, which is a situation which could lead to some serious effect and symptoms. For sure, a more directed approach to targetedly neutralize the bad bacteria could solve this problem. And one approach to do this is to use nanomedicine. In nanomedicine, we actually use very, very small particles to directly treat the disease site. And how small would these particles be? Very small. And to grasp this scale, let's use familiar objects, starting with our body and going closer and closer to a basketball, and under high magnification, an egg, even smaller objects like ants, and still we need to go further in to a grain salt, and even smaller than a grain salt is a human skin cell, which looks like this under this magnification, but still we need to get closer. Closer than a human skin cell is a bacterial cell, and around this magnification, when a bacterial cell would look like this, and this would be 1 micron, which equals to 1,000 nanometers, the particles we are talking about should be at this size. And a common misconception is that we are talking about nanorobots that's swimming in our blood and fixing our blood cells. Well, this is not exactly the case. We are more talking about nanocarriers that in fact are very small parcels that could flow in our bloodstream, for example, and accumulate in a disease site. And these would be called nanocarriers. At the disease site, these parcels that hold the drug inside them would release the drug at the proximity of a sick cell, or even enter the cell and then release the drug inside it. Well, it sounds like a wonderful solution. And if I'm not mistaken, one of the vaccines for the COVID-19 actually uses this technology. But unfortunately, these parcels are quite difficult to synthesize. and they require laboratories protocols with a lot of mixing and drying and expensive equipment. So the parcel itself is very expensive and sometimes not worthwhile. And here comes the neat part, the twist in our research. We suggest a different approach to find these kind of nanocarriers in natural clays. And clays are natural rock, natural minerals with very small particle size. And they are found in many everyday applications such as ceramics and cosmetics and even medicine. And to top it all, they are very abundant and very easily obtainable, so they are not expensive at all. And the clay we are working with is called halloysite. It's the name of the clay. It is mined from the earth like any other mineral and looks like an ordinary white powder using the naked eye. But under higher magnification, very high magnification, you can see here, this is 200 nanometers, a unique tubular morphology is observed, giving it the name halloysite nanotubes, or HNTs in short. And these tubes could be loaded with drugs, actually, and serve as nanocarriers, just like very tiny cannelloni. This brings us to the next step in the research. So we are going to use nanomedicine to treat a very specific site, bringing the drug to the site, using the non-expensive little cannelloni, HNTs, and we're going to use it to treat bacterial infection, to bring the antibiotics directly to the pathogen, to the target bacteria. And how would that work? Imagine yourself a mixture of bacteria cells. This is how a bacterial cell would look like under very high magnification. And these are the green bacteria, the non-target bacteria, the good guys. And inside this mixture, we would find the red bacteria, the targeted bacterial cell, which we would like to neutralize using our halloysite. This is a schematic illustration of one halloysite nanotube. Looks like a cylinder. And the antibiotics are already inside. In order to illustrate better the concept, let us imagine a more common conflict. Hopefully, most of us are well aware of the conflict between transformers and Decepticons. So let's imagine our loaded halloysite is Optimus with his sword, the Decepticon hunter, which is the antibiotics, and his target is Megatron, of course, and all around are Optimus's friends, which he would not like to harm. If the antibiotics would be released evenly, meaning Optimus would use his sword indiscriminately, he might harm his friends. So first, he actually needs to selectively catch Megatron, uses his Stasi-Scarfs, of course. And instead of Stasi-Scarfs, we use antibodies. Antibodies, well known from the COVID epidemic, are actually biological molecules designed to catch a very specific target. And we attach these antibodies onto the surface of the clay, of the halloysite, so they would attach selectively to the target bacteria. In a mixture, when the antibody modified halloysite would bump into a non-target bacteria, the green cell, it would not bind to it. But only at contact with the target bacteria, the good affinity, a good bond will form, and the halloysite will catch the target bacteria, and then the antibiotics, the sword of Optimus, would be released at the proximity of the target bacteria, and we would neutralize only them. And this is, in fact, the targeted delivery we are aiming for. So let's talk a little bit about the science behind this very nice concept. The research itself is divided into two parts: the investigation of the antibody activity, the cuffs, and the activity of the antibiotics. And let's start with the first part. In order to study whether the antibody we attach to the HNTs are working, we used a mixture of two types of bacteria. The red bacteria are the target bacteria, the bad guys, and the green bacteria are the non-target bacteria. And we use two types of halloysite nanotubes, HNTs. One is modified with the antibody, the test sample, and the control sample are halloysite nanotubes, which are not modified with the antibody. And we mixed these compounds together, let them incubate, and then visualize them under the microscope. And this is how it looked like. A comment, the bacteria themselves have a color because they're genetically modified to have that color that we are seeing. So under the microscope, for the control sample, the micrograph looks like this. A micrograph is a picture that was taken using a microscope, and as you can see, the magnification is quite high. The black dots are actually the particles themselves, the HNTs, which are schematically illustrated here. And the red cells are the target bacteria, while the green cells are the non-target bacteria. And you could see that all compounds are just evenly dispersed in the mixture, and no type of bacteria prefer to be at the proximity of the particles or not. However, when we look at the mixture with the antibody-modified HNTs, we clearly see that the red bacteria, the target bacteria, are clustered together with the black dots, meaning that they prefer to be at proximity with these HNTs. So the antibody catch the bacteria, and so the bacteria are clustered together with the particles. At higher magnification, these aggregates, these clusters look like this. You could easily see the bacteria cell clustered together with the HNTs, and you can see here the mouth of the tube and the tube aggregates. These kind of images at extremely high magnification is obtained only by a very special kind of microscope, not a light microscope, but an electron microscope, which uses electrons instead of light to image the samples. And this is a complex instrument which needs a whole room to operate. So this is in fact was the first part in which we showed that the modification with the antibody work. We can catch the target bacteria, but up till now there was no antibiotics at all. So let's talk about the effect of the antibiotics. In order to load the antibiotics inside our HNTs, we merely mix them with a very concentrated solution of the antibiotics. And so the antibiotics goes inside the tube, and the antibodies are already attached to its surface. And then when we mix these antibiotics loaded HNTs with the target bacteria, we expect them to release the antibiotics only at the proximity of the target bacteria. And under the microscope, it looks like this. Again, we see the familiar clusters of cells with the particles, but now we need to investigate the effect of the antibiotics. Do they really neutralize the bacteria? And bacterial growth is measured by measuring the opacity of the sample. Why? Because bacterial cells reflect light like any particle. So when there is no growth at all of bacteria, over time, there will be no change in the opacity. The sample is just translucent. And this is a control sample with no bacteria at all, only growth medium. Another control is a sample with growth medium and bacteria. And here we can see that over time, the sample becomes more and more opaque, meaning there is bacterial growth. But when we mix the bacteria with our antibiotic-loaded HNTs, we can clearly see that the growth of the bacteria is inhibited, meaning that the antibiotics that the particle release indeed neutralize our target bacteria. And for sure, there's still much more to do. We need to investigate whether this effect is in fact really selective, and do we neutralize only the target bacteria and not all the bacteria in the mixture. But this is for the next step in the research. In the far future, could be this kind of preparation will be used to treat, for instance, gut infection, where the antibody-modified halocyte will catch the target bacteria at the inflammation site and release the antibiotics only there, neutralizing the bad bacteria and leaving the good bacteria alive. And with this optimistic tone, I would like to conclude this short talk, So we talked about the need for targeted delivery that could be solved using nanomedicine, but for which the particles themselves are sometimes very expensive, and maybe natural clay that looks like very tiny cannelloni could be a very good alternative for this from an unpredictable out-of-the-box solution because it is very low cost. And finally, we try to use these modified clays to treat selectively pathogen bacteria. So thank you very much for your attention, and I hope this kind of interdisciplinary research would have its impact, maybe on the scientific community in general. But at the time, the effect is mostly domestic. As you can see, this is an art of work my 3-year-old brought from the kindergarten, probably inspired by my research with the bacteria. So thank you very much. And I would be happy to answer some questions.
So one thing I'm not sure, I mean, you sort of discussed it, but I wasn't clear about the rough timetable about whether, like, when do you think this will become available for even not humans, but just like 20 years? Is this a 20 years technology? Is this like a 10 years? That's when you finish your PhD.
Well, based on my experience, I have 5-year experience in the pharmaceutical industry. I wouldn't bet on actual drug coming out of this kind of preparation, but maybe a more like a generic platform. There is some kind of challenge to internalize these kind of materials into the body, but maybe some kind of topic preparation for skin or, yeah, mostly skin. In the skin, we also have a microbiome we would like to protect. So maybe this would be some kind of future applications that for a real drug, it's even up to 20 years. But I really hope that at the end of my PhD, right now I'm kind of in the middle, I would have some kind of, let's say, hope, and maybe using live cells and bacteria. And of course, there's the whole part of toxicity that had to be investigated. More or less, the clays are biocompatible because they are natural and are non-toxic mostly, but all the chemical modifications we do to them in the process, which I did not go into, had to be investigated for its toxicity. So I hope I answered that more or less.
I have a question, please.
Mm-hmm.
First, this is the native structure of the tubes, or you need to go do some process for them, for the clay? Mm-hmm. And the second one is, how is the affinity for with the antibody loading to the tubes? How is the affinity between them, and what release the antibody when it's in the bonding to the bacteria? Or what is it? Mm-hmm. Because if it's good affinity, then you need to release it at some yes. point. But how it's happening?
Okay. So first of all, the structure of the tubes is completely natural. This is how they come from nature. Geological processes just grow these layers of it's alumina and silica. They are all grown together naturally because of nature. And second of all, I can show some slides for that. I can go a little bit through the chemistry. It starts with chemistry of silica. We do some selenization with amino groups and then conjugate the protein onto these amino groups, and onto this protein A, it's a protein with a very high affinity to antibodies. The antibodies are immobilized in a proper orientation. And one thing I really need to emphasize that the antibiotic effect is not due to the antibodies. The antibodies are only a capture probe that is designed to, for the particles to catch the target. They're immobilized onto the surface and catch the target bacteria, and the antibiotics are then released from the lumen of the tubes. And this is the antibiotic effect.
But how they can be released? Only when they attach it to the cells. This is my question. Yeah.
Okay, this is a good question. This is also a next part of the a possible part of the research. We may use some triggering mechanisms, such as a layer of biopolymer that will be degraded by the bacteria only when they're at their proximity. Right now, we are only based on the fact that the antibiotics is released slowly because it is contained inside the tubes. It has controlled release, sustained release. So it is released slowly, and we hope that first the binding occur, and then the antibiotics is released slowly at the proximity of the bacteria.
It's diffusion or something like this.
Yes, it's diffusion and hinder diffusion because it's inside a very small tubes.
Thank you very much.
I have a question if there's still time.
Mm-hmm.
So ultimately, though, this kind of hinges on antibody specificity, right? For the nanotubes to even be able to recognize the right bacteria, but I imagine that still leaves open a whole lot of potential bacteria which could be misidentified. Is there, like, what sort of how do you approach that problem once you scale up from beyond to kind of?
In our research, we used E. coli as a target bacteria as an example, and the antibodies were purchased as anti-E. coli antibodies. In general, at this currently, antibodies are the most specific targeting mechanism available right now biologically. It could recognize some other Enterobacteriaceae, but as you see in this case, it was E. coli and Listeria, but only just for example. We do think about the future, mostly because antibodies are expensive. We think about aptamers, if you have heard about them. They are very short sequences of DNA that could reform to have secondary structure and then recognize very specific targets as well. But it's a little bit problematic because they are mostly targeted against molecules and not against cells. So aptamers against cells is a quite new area. We tried it, we failed, but still it's very easy to try many kinds of aptamers. And there are more kinds of proteins like wheat protein that have affinity to bacteria, but then it's more general and less specific. So right now, antibodies is all what we have, but maybe, hopefully, aptamers could have a better option in the future.
Thank you.
Okay, thank you very much for hearing this cookie research.
Actually, it was very interesting.
We have Ofer, we have 5 more minutes. So if anyone has a question, now will be a good time to ask before going back to the main session.
To be honest, I think every question I want to ask, someone that just asked it before me. So just want to say it's a really nice job and nice work. And well, you talked about it, about using antibodies and why you should replace antibodies. But did you have any idea how antibodies will survive all the way to the guts? I mean, it's a very hostile environment for proteins, especially antibodies, which needs to keep their structure. So do you have any knowledge on it?
It is a very good question. For sure, it will have some effect on the stability of the antibodies. The delivery into the gut, up to the gut, down to the gut actually, is something that pharmaceuticals have already solved. There are all kinds of polymers that degrade only at the pH of the gut. It's called the Udrigit, and it's a combination of methylacrylat and metamethylacrylat that you can play with on the backbone of the polymer. So maybe in some kind of capsule, it will go only down to the gut, and then the antibodies will actually have to be stabilized at pH around 9 or something like that. Yeah. There is some kind of option to stabilize the mobilization of the antibody with some chemistry between the proteins. And as we said, we were really open to find other ways to use other capture probes other than antibodies, and also investigate other administration routes, such as topic or otic for ears, or nasal for nose, or something like that.
So I know one field that's getting really nice attention, or really good attention, is nanobodies, which are basically just a part of an antibody, but can be manufactured by bacteria. So it's supposed to be cheaper, and its structure is much simpler. So maybe this could be another
nice
direction.
Nanobodies. Thanks. Okay, I hope I didn't speak too fast.
It was super interesting. Thank you.
Okay, if we don't have any more questions, we can go back. You can press the leave room and go back to the main session, and then we're going for a break and reconvene again. Ofer, thank you so much. It was very interesting.
Thank you. Thank you for hearing.
Thank you very much.
Thank you.
Thank you.